groups, and phosphate groups gives it a total negative charge. And the phosphate group jumps out of the DNA structure, and that is why it is known as deoxyribonucleic acid. And why is it deoxyribose? It is deoxyribose because it has the ribose sugar at the 2' position. The OH group is replaced by a H group. So you start recapitulating how the DNA looks like first. So this was the basic structure of the DNA, and what happened? This bases were held together by the hydrogen bonds, and you had other interactions between the two strands. You had your van der Waals interaction. You have your base stacking interactions, and these bases were planar. So they were placed on a single plane. Okay? And what happened if you have various natural agents which could actually break the hydrogen, you have got the denatured DNA. Okay? So the denatured DNA you got from using chemicals on the normal DNA. So what were the chemicals, if you remember? They were acids, they were alkalis, and another was the heat. Heat could actually unwind the two strands and given you two single strands. And these two single strands, if the temperature was cool, these two single strands came closer together and again we can rewind it to give you the double helical structure. So this is what we remember about the structure, and there were actually three types of DNA: the A DNA, the B DNA, and the Z DNA. Among them, the A and the B DNA was the right-handed DNA, and the Z DNA was the left-handed DNA, and the B DNA was known as the Watson-Crick DNA, because it was this DNA that the Watson and Crick had seen in their X-ray crystallography and elucidated this structure. So this is a part of your recapitulation of how the DNA looks like. And another thing you remember about the DNA, that they had a typical pitch, they had a typical the typical distance between the two bases, and they had the major groups, they had the minor groups, and what else did they have? They had the helicity, they had the major groups, they had the minor groups, they had their pitch, they had their distance between the two bases, number of bases per turn, that is their pitch. So they had this particular typical characteristics which distinguish the A DNA from the B DNA to the Z DNA. So this was the basic structure of the DNA. So it is from the single-stranded, the DNA went into the double-stranded. Now from the double-stranded we will go to the nucleosomes. So what is the importance of nucleosomes, do you think? Just imagine the DNA. Okay, so that DNA, the whole DNA of your body has been condensed, recondensed to enable itself to be accommodated in such a small organelle of the cell, that is the nucleus. The cell itself is microscopic, and this nucleus is much smaller than it. And imagine how a DNA strand of the total cell has been able to accommodate itself in the nucleus. Again, when replication, transcription occurs, this nucleosome, what does it do? It then opens up and helps the replicating enzymes at your DNA polymerases to come and start replication. It allows the RNA polymerase to come down to carry out transcription. So there are processes of condensation. There are also processes of just going in the opposite sense of the condensation. So you have a condensation and then the opening up of the condensation to facilitate the processes of the body of the cell. So it is so intriguing if you see the structure of the nucleosome. So first of all, we will go into how much DNA a single organism has. into the DNA of the cell. The haploid human genome contains approximately 3 billion base pairs of DNA packaged into 23 chromosomes. This is must be knowing that we have 23 chromosomes in the haploid genome. So you have 3 billion base pairs. Of course, most cells in the body except for the female ova and the male sperm are diploid. So with 23 down to, it comes to 6 billion base pairs of DNA per cell. Because each base pair is around 0.34 nanometers long, a nanometer is 1 billionth of a meter. Each diploid cell therefore contains about 2 meters of DNA. So 0.34 10 to the minus 9 into 6 into 10 to the power of 9. So this is the amount of DNA you have in the cell. Now, moreover, it is estimated that the human body contains about 50 trillion cells. So, which works out to 100 trillion meters of DNA per human. Just imagine the amount. You have this amount of DNA in your body. 100 trillion meters. Now consider the fact the sun is 150 billion meters from the earth. So, what can you do with your DNA? You can go from here to the sun and back more than 300 times, or around the Earth's equator 2.5 million times. This is the amount of DNA you have in your body. And just imagine the process carried out by nature so that it can be accommodated in your cell, in your body, and nothing is damaging out of you. Yes, this is so unique. This is just mind-blowing. So if you go into the organization of the eukaryotic chromosome, this is actually a picture, a schematic picture of how the organization starts. So you have DNA wrapped around the histones. Then this DNA wrapped around histone is known as the chromatin. And then this nucleosomes are called, from nucleosomes or chromatin, and they have appearance of beads in a string. Just you can see the third part of the third window of the figure. Then you go down to further condensation of chromatin, and then you have the duplicated chromosome. So now we will go into the details of how this type of organization is taking place and who is helping the DNA to get complexed into such beautiful structures and how this is occurring. First we will go into a brief overview of how it is occurring. Then we will go into the scientific discovery of the people who have done it before we go into a detailed explanation. So the answer to this question is, certain proteins compact DNA into the microscopic space of the eukaryotic nucleus. So DNA is what? DNA is negatively charged. So the proteins which will be coming and binding to it will be positively charged, of course, otherwise why should it bind? A negatively charged protein will not bind to a negatively charged DNA because they will rebel. So the first and foremost criteria of those proteins which will be coming to bind this DNA, so for compaction, should be positively charged. These proteins are called histamines, and the resulting DNA protein complex is called chromatin. Okay, and it may seem paradoxical that proteins are added to DNA to make it more compact. DNA itself can go for a double helical structure, and you need proteins. Proteins are large molecules for compaction of the DNA. So, however, if you have ever tried to store a garden hose, you know that it is much easier to do so if you begin by coiling the hose. Yes. So this is the same principle which is being applied to the DNA coiling. So it is easier to coil it, but you need energy, isn't it? You give energy for coiling of the garden hose. Coiling requires work and energy is needed to perform this work. From where from does this DNA gets this energy? So as to coil itself. So how does it coil itself? The protein binds and the protein helps in its coiling. Thus within the nucleus, histones provide the energy maintained in the form of electrostatic interactions to fold the DNA. So positive charge, negative charge, there's a huge electrostatic interaction and it helps to fold the DNA. As a result, chromatin can be packaged into a much smaller volume than the DNA alone. Yes, so these histones have a very important role in packaging the DNA. So if histones were not there, the DNA would have been dangling here and there. So the histones have the capability to package the DNA, and you have 5 types of histones as we will be discussing. These 5 types of histones are so much responsible for packaging the DNA. So now we will go into the organization slowly. Histones are a family of small positively charged proteins termed H1, H2A, H2B, H3 and H4. Van Holde had discovered them in 1988. They had gone for the elucidation of the nucleosome structure, and when they had gone for the elucidation of the nucleus structure, they found that these were the proteins which were actually helping in the compaction. DNA is negatively charged to the phosphate groups in the phosphate sugar backbone, so histones bind with DNA very tightly. This nucleosome is linked to the next one by a short strand of DNA that is free of histones. So if you can go back to the structure, you will see that in the third window you can see the nucleosomes, and in between the nucleosomes what you see? You see a small part of the DNA which is not bound by the nucleosome and is free of the histone proteins. So that part is the DNA which is known as the linker DNA. So DNA connecting the nucleosome is called the linker DNA, and this is also known as the beads of a string structure. The nucleosomes are the beads and the short lengths of the DNA between them are the string. So something looks like a necklace, isn't it? So you can actually compare between the necklace, and you will find that it's much more similar to a necklace. So it's known as the beads of a string. And this is known as the beads of string structure. The nucleosomes are the beads and the short lengths of the DNA and the string. And a DNA molecule in this form is about 7 times shorter than the double helix without the histone. So, and the beads are about 10 nanometers in diameters, in contrast with the 2 nanometer diameter of a DNA double helix. So you just imagine the amount of condensation that is taking place. It is being shortened 7 times in a single go. Okay? Now, we will go into a detailed study of the nucleosome, that is the DNA, the histones packed together. The basic repeating structural and functional unit of chromatin is the nucleosome. So we will find out now who had discovered and who has actually contributed to this study of packaging, which contains 8 histone proteins and about 146 base pairs of DNA. Mind you, there are 5 histone proteins. Yes, we just now studied. So amongst the 5 histone proteins, you have 4 histone proteins out here. So you have H2A, 2B, H3 and H4. As you will go into the structure, we will see that H1 will come a little later for the packing. And 146 base pairs of DNA and 8 histone proteins. Okay. Then this observation by electron microscopes that chromatin appeared similar to beads on a string and provided an early clue that nucleosomes exist. It was done by Ollens in 1974 and Woodcock et al. In 1976, and that 146 base pair of DNA with histone proteins, 8 histone proteins, so 4 histone proteins, so they are coming as dimers. So it was done by Van Holden 1988 and Wolfe in 1999. So if you are very much interested, you will find their papers, if their research papers which have got published, by searching them by their title. Another clue came from chemically cross-linking histones in a chromatin. So it was done, cross-linking is joining the histones actually, and it was done with Thomas and Kornberg in 1975. Kornberg, I think, also got a Nobel Prize for this nucleosome structure. The experiment there was H2A, 2B, H3 and H4 from a discrete protein octamer. So you know what is octamer? Octamer is a structure where you have 8 subunits, which is fully consistent with the presence of the repeating histone containing unit in the chromatin fiber. So here you have a electron micrograph picture of the beads on a string. Okay. Now today researchers know that nucleosomes are structured as follows. To each of the histones, H2A, H2B, H3 and H4 come together to form a histone octamer, which binds and wraps approximately 1.7 turns of the DNA, that is 146 base pairs. The addition of one H protein wraps another 20 base pairs. How many base pairs do you have? Now 166 base pairs, resulting in 2 full turns around the octamer. Okay, and forming a structure called chromatosome or nucleosome. So just recapitulate what you have got. So you have histone proteins, you have DNA. How many DNA? First the octamer of histones wrapping around 146 base pairs. There you have the one condensation, that is 1.7 turns. In comes H1 protein, histone proteins with another 20 base pairs, giving you 166 base pairs and 2 full turns around the octamer. The resulting 166 base pair is not very long. Consider that each chromosome contains over 100 million base pairs of DNA on average. Therefore, every chromosome contains hundreds of thousands of nucleosomes. Yes, and these nucleosomes are joined by the DNA that runs between them, and the linker DNA, it has been found experimentally that it is about 20 base pairs. So now you can visualize up to the nucleosome. Now we will go for another further condensation. Yes. So if you see this structure, this is total packaging of the DNA. So first of all, step by step, and then I can study and come back to this picture again. So first is that at the so first you see the DNA double helix with 2 nanometer diameter. Okay. At the simplest level, chromatin is a double-stranded helical structure of the DNA. Got it? Second step. DNA is complex with histones to form the nucleosomes. So nucleosome core of 8 histone molecules. So you need the solenoid. You remember your solenoid. So this is the coiling of the DNA around the octamer. Each nucleosome consists of 8 histone proteins around which the DNA wraps 1.65 times. Fourth, a chromatosome consists of a nucleosome with the histone protein. So this much we have studied. So this much should be clear with you. Just let me know in the chat box that up to this much it is clear with you. Then we go. So this is the picture's description. You can go for it later. So the chromatin now is coiled into higher order structures. The packaging of DNA into nucleosome shortens the fiber length about 7-fold. In other words, a piece of DNA that is 1 meter long will become a string of beads chromatin fiber that's 14 centimeters, about 6 inches long. Okay. Then despite these shortenings, a half foot of chromatin is still much too long to fit into the nucleus. So it will have to go for a further condensation, which is typically only 10 to 20 microns in diameter. The nucleus cannot accommodate such a long DNA, so it will go again for a condensation. Therefore, chromatin is further coiled into an even shorter, thicker fiber, termed the 30 nanometer fiber, because it is approximately 30 nanometers in diameter. So if you go to the picture, you will find the next step. The nucleosomes fold to produce a 30 nanometer fiber. So this is how it looks like. Okay? Step. Interest in H1 is very important in stabilizing the chromatin high order structures, and 30 nanometer fibers form most readily when H1 is present. You understand? So now we will go that how does the processes which such as transcription and replication require the two strands of the DNA to come apart temporarily, thus allowing the polymerases access to the DNA template. DRC sir has gone with you in this process, in this study, and you know that for transcription and replication you need the DNA to unwind, and it unwinds from the nucleosome structure. It does not unwind from the double helical structure. If it had unwound from the double helical structure, it would have been so much easier, but you have to open one by one or of the packaging. So you just imagine if you have a gift box which is packaged with 5 papers and you want to see what is in the gift box, you have to open the wrapping one by one. So here the same thing takes place, so you have to open it up. However, the presence of the nucleosome and the folding of the chromatin into a 30 nanometer fiber pose barriers to the enzyme that unwind and copy DNA. It is therefore important for cells to have means of opening up chromatin fibers and removing histones transiently to permit transcription and replication to proceed. So can anyone tell me what are those proteins or enzymes which help in this unwinding and removal of the histones so that transcription and replication takes place? So we go to the structure back again. So from the 30 nanometers fiber you will have now 300 nanometer. Okay, 300 nanometers. So how does it occur? Comparing the length of the metaphase chromosome to that of a naked DNA, the packing ratio of DNA in metaphase chromosome is approximately 10,000:1 depending on the chromosome. So if we go back to the structure again, so from the 30 nanometer chromatin fiber which forms loops averaging 300 nanometers in length. So this is the part of the 300 nanometers. So from 30 nanometers it goes to a 300 nanometer loop-like structure, and then the 300 nanometer fibers are compressed and folded to produce a 250 nanometers wide fiber. So how does it look like? So it looks something like this. So from step 5, the nucleosome folds up to produce a 30 nanometer fiber, and from there it goes that forms loops averaging 300 nanometers in length. So this is the 300 nanometer loops, and then from the 300 nanometers loop it goes compressed and folded to produce a 250 nanometer wide fiber, and the last coiling is that tight coiling of the 250 nanometer fiber produces the chromatid of a chromosome, and that is what you see actually, the chromatids. Yes, where do you see? During cell division. So they have a 1400 nanometer. Just imagine, if you see from the first part of the figure to the last part of the figure, see the amount of coiling and condensation, and that is how nature works. So the level of compaction is achieved by repeatedly folding chromatin fibers into a hierarchy of multiple loops and coils. Exactly how this is accomplished is unclear, but the phosphorylation of histone H1 may play a role. And chromosomes are most compacted during metaphase. When eukaryotic cells divide, genomic DNA must be equally partitioned into both daughter cells. To accomplish this, the DNA becomes highly compacted into the classic metaphase chromosome that can be seen with a light microscope. Once a cell has divided, its chromosomes uncoil again. Okay? So because it has to uncoil, because it has to be given to the two daughter cells. So if it doesn't get uncoiled, how will it be divided and segregated into the two daughter cells? Okay.